I'd like to discuss here a basic biological and physiological phenomenon known as osmosis. Now, we discussed diffusion in class already, and diffusion is the movement of molecules throughout any kind of solution from an area of high concentration to an area of low concentration. This principle is one of the driving forces behind some of the most complex physiology we discussed in Human Anatomy and Physiology 1, but you also discuss this in Biology 1, and it's fundamental to understanding some basic life processes. Now, the important concept to remember is the high to low concentration. In this case, I want to discuss the movement of water. Now, osmosis has to do with the movement of water. So, imagine here I'm going to draw a beaker, and I have a little beaker, and I'm going to, in that, divide this beaker into half. Now this beaker is semi-permeable. I have a membrane between this beaker that's semi-permeable, and what I'm going to do is I'm going to come into this beaker and I'm going to put water in it. Now, we can put water at different levels here. Now, for us to kind of imagine is what's going on here, imagine if I were to come by right now, you would say that the water concentrations are equal on both sides. I'm going to give you a 1 and a 2, so we can discuss these in terms of side 1, side 2. Right now, side 1 and side 2 are equal to each other. Water molecules will move equally in and out and back and forth in equal distributions, and they will have no problem. There will be no change in how these guys will move. So they're going to be pretty well equal. So what I'm actually going to do, just to make things a little more interesting, is I want to mess with the concentrations. Now, water is the only thing that actually moves in osmosis. So imagine if I come in and I sprinkle on side 1, or side 2 here, some salt, so that I have salt in here. That salt, now there are salt molecules in this side. So now you would say side 2 has a high concentration of salt, or to say a low concentration of water. Water will move from an area of high concentration to an area of low concentration. Now that there is high salt, there is low water, H2O. This side is high H2O. So imagine, if you will, what's going to happen to water. Well, side 2 needs to equalize. If we want to get to equilibrium, we can move water to this side, and the water level on side 2 can rise to equalize these two sides, give them an equilibrium. That means side 1 will drop. So I can drop the water level there, raise the water level there, and now I actually have a beaker whose osmosis will change. And then if I flip this around, it's the same basic principle, high to low. And you can use this basic idea for, but this is only for water, but this is used in Biology 1 and AP 1. So now we have the water dropped here, but raised on side 2. If I flipped it around, the same thing is going to happen. It could be glucose. It can be any other thing. So I'm going to give you, now that we've had a beaker, let's give you just a simple biological example as I erase some of my mess. And let's give you a cell. And I have a blood cell here. So this is a little blood cell and erythrocyte. Now, outside here, we're going to have ourselves glucose. And it's going to be high. Now, what does it mean if glucose is in high concentration? If glucose is high, water is low. Water moves from an area of high concentration to an area of low concentration. Well, the glucose level here we could definitely assume if water is in low concentration, it is low with respect to the inside of this cell here. That means that in order to get this water in a low concentration, we want to get more water in there, we shift the water out. This cell will shrivel. So if this cell is in a solution that we call hypertonic, it shrivels. Now, let's imagine what's going to happen again here. Let me erase this. Let's draw our blood cell. Very rudimentary, very simple, very crude. But now we have glucose, and it's low, because in the blood cell it is high inside. Well, the water will need to move into the cell, causing it to burst. If its glucose is low, we can call this hypotonic. And this results in the bursting of the cells. And this is similar to what happens in a diabetic. And this is why you end up losing tissues, why blindness, why you have diabetic neuropathy, nerve damage, all these sorts of problems. If we have a high concentration of glucose gets in our blood cells, we start damaging tissues, and we start causing all kinds of stuff when we have high glucose levels. So in our blood, when we have that problem, this will cause cells to burst, and we have a lot of issues dealing with this. So we would see this, this is, osmosis has some very important stuff. It's also diabetics. It's one reason why there's another cause with the lens, with water leaving there that can result in blindness as well. So the hypertonic. Now, just one simple thing as I clear one more time. The simplest case, which I could have done first, we have a blood cell, and glucose equals that in its isotonic. Isotonic, you will get the same amount of water going in as going in and goes out. It will stay the same. And so this is a pretty basic idea, but this can be as fundamental as trying to explain why the chemistry of life requires water to dissolve these ions in. And simple diffusion explains how nerve cell conduction works, but this is a very simple idea. And I thought I'd give you a very simple case on how this works, very rudimentary, more simple than what you guys can see on the board. But, as we look at our PowerPoints, this is definitely crude compared to that standards, but you may find it a very useful thing. And this concludes my little simple video on osmosis.